We are now in chapter 3, and chapter 3 is about the cells. Cells, as we saw in chapter 1, are considered the basic unit of life. This is the tiniest or the smallest unit that sustain the properties of life. And almost every function in our body is performed through cells. The cells are made out of three basic components. We have the plasma membrane, the cytoplasm, and the nucleus. In this chapter, we are going to go through each one of these parts. For the cell membrane, as we saw in chapter 2, it's basically made out of two layers of phospholipids. If you remember the phospholipid, we have fatty acids, two chains of fatty acids, we have glycerol group, and we have a phosphate group. So this is a structure of the phospholipid. For the cell membrane, it's made out of endless repetitive of these structures. One thing that is unique about the phospholipid that it has two parts. It has hydrophobic tails and it has hydrophilic head. Hydrophobic tail is afraid from water or hate water, and the hydrophilic head likes water. And since we are talking about two bilayer of phospholipids, it means that these hydrophobic tails are going to face each other, whereas the hydrophilic head is going to face the intracellular fluid and the extracellular fluid, since it likes water. Now we need to remember that the cell is always surrounded by water. There is the extracellular water, and inside, this is something we will understand in a bit, also there is water. So these heads, since they like water, they are going to turn toward that water, whereas the part that is afraid from water is going to stay in the middle. The phospholipid would make around 70 to 75 of the plasma membrane, whereas in other components will be 5% glucolipids and around 20% cholesterol that would stabilize the membrane. Now, for the plasma membrane, it has several functions. It serves as mechanical barrier. It separates things, ions, particles from inside and outside. Selective permeability, we'll understand what is this. Electrochemical gradient, it separates ions, whether they are negative or positive ions, from inside and outside. Also, it helps in communication and it helps in cell signaling. All of these are functions of the plasma membrane. Now, along with the phospholipids, there are proteins in the plasma membrane, and we have two types of proteins. We have integral proteins, like this one, this one, this one. These are integral proteins, and these are embedded within the membrane. Embedded means that they are part of the plasma membrane. We cannot remove them. If I have to remove this or this protein, it means I'm going to destroy this part of the plasma membrane, which might cause the cell to die. So these are integral protein, meaning that they are fixed within the plasma membrane. The other type is something called peripheral proteins, like this one, maybe this one. All of these are peripheral proteins. These, as you can see, they are not really spanning across the plasma membrane. They are only in one side of the plasma membrane. The integral proteins span across the plasma membrane, whereas the peripheral protein do not. So therefore, these peripheral proteins, sometimes they can leave the plasma membrane and go inside the cell, or they can leave the plasma membrane and go to another cell. And this will not cause any problem to the plasma membrane. So these are the two types of proteins within the plasma membrane. Also, these proteins serve different functions: transport, cell recognition, receptors and ligands, anchors for other cell structures, enzymes, and forming joining areas. We will see some of these functions like the transports, anchoring to other cell structures, forming joining areas. We will see them through this course, whereas others we might see them in EB2. Now, let's see the transport across the cell membrane. We said that cell membrane, one of its function is to form a barrier. And of course, we understand that when we have the cell, it has to get food, for example, from outside. And also this processing the food would lead to waste, and this waste has to go outside the cell. So therefore, this process of transporting materials outside and inside is happening across the plasma membrane or the cell membrane. And therefore, we can say that the plasma membrane is selectively permeable, which means it allows certain materials to come in and certain materials to come out. Not everything, some of the materials will be denied to leave or enter the cell, whereas other materials can come in or come out of the cell. And usually these materials, they are small nonpolar materials that they can go through this lipid bilayer directly without any help. But there are other materials, they need to be transported, meaning that they need a kind of a help, a kind of a tool to move them either toward inside or toward outside. And therefore, sometimes these transport processes would require energy. So we have two types of transportation processes. We have passive transport or passive process where there is no energy required. And the second type is the active process or the active transport where energy in form of ATP is required. So these are the two types of transport. And each one has subtypes. For the passive transport, we have diffusion. Sometimes we will call it simple diffusion. There is facilitated diffusion and there is osmosis. On the active transport, there is the active transport itself. Then there is something called endocytosis and something called exocytosis. Now, before we go into the mechanisms of transport, we need to understand the concept. That is a concentration gradient. Concentration gradient, if you are looking at this image, you understand that this dye is trying to diffuse or move to the area that is undyed yet. And if we look here, we can see there is a higher color or a darker color of the dye. At the same time, if we move away from this area, we will see that the color is declining a little bit. Like here, for example, this area is lighter than this area. This area may be lighter than this area, and so on, right? So here we can see that there is a gradient of colors in this particular example. This gradient of color refer to different concentrations of the dye. The concentration of the dye here is less than here, and here it's less than here, and here it's less than here, and so on. So this difference of concentration across this area is called concentration gradient. So the concentration gradient is the difference in concentration of a substance across a space. You can think of it as a stair. Every time you go down, the concentration is diluted, okay? The concentration is less. So this concentration gradient caused the substance to move from the area of high concentration to area of low concentration. Again, because of the concentration, because of the high concentration in this area and the low concentration of this area, so the dye is going to move from the area of high concentration to the area of low concentration. And this is something called moving down their concentration gradient. Moving down their concentration gradient. So as you can see here, the dye, we start at certain point and the dye goes all the way to fill the cup. It's moving from area of high to the area of low concentration. This is the movement down their concentration gradient. Now in the cells, and because of the plasma membrane with its selective permeability feature, it always creates a concentration gradient for several particles. And these particles, they will try to diffuse inside, but they need a time. So there will be a time before we have equal concentrations of these materials across the plasma membrane. And in fact, in many times, it will never happen that there is equal concentration of materials at both sides because of the plasma membrane. So the plasma membrane, that's what does it mean that the plasma membrane would create a concentration gradient, a chemical gradient, because it's blocking the materials from moving inside toward the area of low concentration. So the first form of transportation across the plasma membrane will be the diffusion, the simple diffusion. This happens, for example, for oxygen and CO2. Oxygen and CO2 that can easily diffuse across the plasma membrane. And this is simple diffusion because, again, it does not need any aid. They directly diffuse. And of course, it's a passive transport. It means there is no ATP needed for this transportation. Now, let's talk about the facilitated diffusion. As you can think of the word facilitated, it means it happens with aid or it happens with the help. So here, there is a help for this diffusion to happen. Still, we are in the passive transport, so there is no ATP needed, right? But we need something to help these particles to move across the plasma membrane. Sometimes their size does not help, their charge does not help, their polarity does not help. So we need something to move them in, and that is going to happen through facilitated diffusion. Diffusion, remember, also it means that they are moving down their concentration gradient. It means that they are still moving from areas of high to low. That is the concentration gradient. That is how they are moving. They are moving along their concentration gradient. So for the facilitated diffusion, we have two forms. Either through channel proteins, like this, for example, channel proteins. Usually these are always open. Sometimes they will close, but in general, they are easy to open, easy to close. And once the particles arrive, they will simply go through them. An example of these, like there are channels that would transport water. As water arrives, it will move directly through them. There are also some ions that would be transported through this way. So this is the channel protein. The other form is carrier proteins. And for carrier proteins, they are a little bit different than channels. Channels, you can think of the channels that as like a tunnel. You will go through it. Whereas carrier protein, you need to think of them as like elevator or a crane, that it has to pick the material from one side and turn to put it to the other side. So there is kind of a shape change in this process of the carrier protein. So for example, it will pick this material, it will change its shape, then to release it from the other side. So here, the rate of transportation is dependent on the availability of these carrier proteins. Whereas with the channel proteins, it's always there. Higher concentrations would mean faster transportation or a faster rate of transportation. Whereas here, even if I have extra like couple of hundreds of these particles, no, we will wait for these proteins to change their shape and move these particles one by one. So this is the carrier protein. Now there are like sugar, amino acids, plenty of ions that are transported through the carrier proteins. Remember, all of them, they are moving from areas of high to low, and there is no ATP used in this process. So this is the facilitated diffusion. So I will stop here. When we come back, we will talk about osmosis.